centuries, people gazed at the living world and wondered how it began. Did life emerge from the inert, as alchemists once claimed, or was every creature born from another? This question haunted philosophers and scientists alike until experiment replaced speculation. In medieval Europe, believers in spontaneous generation argued that life could spring from non-life, maggots from meat, mice from grain, microbes from broth. Others, skeptics like Francesco Redi and later Louis Pasteur, devised careful tests showing that even microbes have parents. Their flasks of sealed broth remain sterile unless touched by life. The conclusion was profound. Every living thing descends from something alive. Modern evidence from geology, chemistry, and genetics extends that truth far deeper. All life on Earth, including the smallest bacteria and archaea, arose from pre-existing life, and microbial life itself appeared astonishingly early. Fossilized microbial mats called stromatolites, preserved in rocks over 3.7 billion years old, bear the fingerprints of ancient metabolism. Life began almost as soon as the planet cooled from its cosmic birth. The early Earth was no paradise. Beneath a young, unstable sun, volcanic gases filled the air, and the oceans simmered with dissolved metals and lightning storms. Yet within this chaos, chemistry learned to organize itself. Molecules that could store energy, form membranes, and copy information began to collaborate. These self-organizing systems, the ancestors of cells, turned chaos into pattern. Modern experiments echo that ancient transformation. In controlled flasks, simple gases such as methane, ammonia, and hydrogen can yield amino acids and nucleotides. Mineral surfaces act as catalysts, concentrating and protecting fragile molecules. Some researchers propose that life began in deep-sea hydrothermal vents, where gradients of heat and minerals powered primitive metabolism. Others imagine tide pools, where evaporation concentrated organic matter into protocells. Wherever it began, the recipe was universal, chemistry seeking stability through replication. The first true cells likely resembled modern archaea, heat-tolerant, anaerobic organisms that harness chemical energy from sulfur or iron instead of sunlight. Over time, these primitive microbes diversified, adapting to the planet's shifting atmosphere and temperature. When cyanobacteria evolved oxygenic photosynthesis, they transformed the world, filling the air with oxygen and paving the way for complex life. The Great Oxidation Event, once a catastrophe to anaerobes, became the foundation of evolution's next act. Evolution itself began with these microbes. In experimental systems, new bacterial colonies evolved visible differences in just days. Wrinkled surfaces, faster spreading, novel pigments. Every mutation is a tiny replay of life's origin, showing how random change and environmental pressure sculpt diversity. Natural selection is not a distant theory. It unfolds in every petri dish. By tracing molecular fossils, genes, proteins, and ribosomal RNA, scientists have uncovered deep connections among all organisms. Every living thing, from archaea to animals, shares the same genetic alphabet, the same energy currency, ATP, and the same molecular logic for translating DNA into life. Such unity implies a single common ancestor, LUCA, the last universal common ancestor, a microbial progenitor that gave rise to all known life. From dust and starlight, from heat and chemistry, life wrote its first sentence billions of years ago. Each microbe today is a word in that ongoing text, evolving, adapting, diversifying, a reminder that the story of life began not with complexity, but with persistence.